who were not here, I am checking those and putting those in. So that DNA template, you should have the mRNA codons that match up to it. You should have the TRNA that goes along with the anticodons that complements them. And then you should have the protein structure that is made from there. Any questions?
You have a copy of that I can get?
I think I have an extra copy right there on top of that giant stack of papers. Yes, you don't have to do the coloring and the activity at the bottom, just the coding out and making the mRNA, tRNA, and the protein. So, good to see you guys today. Good to see everybody back, except for our one we're missing here. Hope you're all doing well. Let me emphasize this, and for the 3 people out there on the internet, we are going to get into stuff that's way more complicated than what we've been over so far. And I know DNA replication and protein synthesis was pretty complicated, and there's a lot of enzymes and sequences going on in those different concepts. So I need you to be on your A game and make sure you are studying and looking over the things that you need to study and look over in order for you to be successful. I think yesterday I got to finish passing out your fee sheets for the AP exam, but I will pass those out momentarily. Those are some pretty hefty numbers if you just look at them, and I know your parents probably freak out when they see those fee sheets, but you got to keep in mind that an AP exam costs about $90 for the test, and they grade it, and a class in college, college level class right there, it's probably closer to $1,000. If you just pay tuition for one, I think, let me see, 1,000, class you get 5 or 6, yeah, that's about right. So maybe even a little bit more at the bigger universities. So, and as well noted behind me here, the bio pass rate's pretty high historically. If you do what you're supposed to do, you're studying, you're reviewing, you're asking good questions, you're paying attention, you're printing off your notes and reviewing over them, doing what you're supposed to do. So if you will do your part, I will make sure that you have the information and things that you need to be successful. You probably picked up on this after a half year in my class. I'm not a micromanager. I do not give tons of work outside of class to keep you on point. I try not to give much homework at all, but I do expect you to read over your notes, to study, review, and to make sure that you're understanding the things that we talk about in class. So, which probably looks more like what you will have to deal with in college, Ashlyn, in the future. So I'm hoping you guys get used to that. I don't have a lot of confidence on online learning because I think it's hard when you're at home to be self-actuated, especially the younger you are. I know it's hard for a lot of college students to attend their online classes, much less, if you're in high school, or I can't imagine what middle or elementary school is like. So anyway, last thing to note, Walker, I want you guys to know that I am here for you. If you're having trouble, write down your questions, come see me. If you need to talk about something, I'm here. If you need a reference for a scholarship or something, 1, don't be a slacker, and 2, make sure you send me all the pertinent information, and 3, don't ask me a day before you need it, because that's inconsiderate, okay? But I'm here to help you be successful and do anything I can to help in that endeavor, okay? I definitely think all of you have a good shot at passing the AP exam. And bio is, like at the university, counts as 2 courses and 2 lab classes, which is roughly half a semester's worth of college. And regardless of whether you're going anything sciencey or not, most colleges require 2 science courses. So for some of you people who have decided that science is not for you, whatever. I would suggest you get these science courses out of the way then, if that's your objective. If something in science, especially biomedical, somewhere in that range, is for you, then I would be paying attention because these are all the basics we're going over. Okay? So, I'm here for you. If you need something, let me know. I have high expectations for you guys, but quite frankly, some of y'all have been slackers, and now's the time to pick up the pace because in the spring, genetics, ecology, and evolution probably make up about 60% of your test. So now's a good time to make sure you're paying attention and understand what's going on, okay? And there's a lot of details in here, but guess what you need to know details for, Fife?
AP exam.
FRQs, good. Some of y'all might wing some of the multiple choice because you're good at data analysis, but you need to know details to write information out for the FRQs, okay? So let's get started. Did anybody have any questions on protein synthesis part 2? Fortunately for you guys yesterday, I think we were in here and y'all got to finish that up and we talked about it and elaborated on all things protein synthesis, page 354 in your book, Walker. This is a very good overview of that process. Let's go through it really quick. 354. And I'll say this, I see Tay writing down some notes there. When you're talking about notes, and I think most of y'all have got your notes printed off, scientific studies show that having your notes on your phone or on a computer or even taking notes on a computer is much less effective than handwriting out notes. So I know that I've given you most everything, kind of the basics from the unit here. But when I ask you questions like, why does this happen? When does this happen? Where does this happen? What does it relate to? These are things that you should think about and ask yourselves. That's how you learn. That's how you remember stuff. So keep that in mind. When you write down, you have to think about things and put that in information down, and it helps you remember. All right, number 1, Walker, 354.
RNA is transcribed from a DNA template.
All right, we went through the intricate steps of the transcription initiation complex, initiation, elongation, and termination. You might want to go back through and look. But the basics are DNA unwinds, RNA polymerase II goes through, adds in nucleotides. In which direction?
5 to 3.
5 to 3, and makes a copy of that DNA. What's the DNA section that it copies?
Gene.
A gene. So it's a segment of DNA that's going to code for a protein. Number 2, in eukaryotes. Mason.
In eukaryotes, RNA transcript is spliced and modified to produce mRNA, which moves from the nucleus to the cytoplasm.
All right. So RNA processing has to take place in eukaryotes. What has to be removed? Introns. Exons have to be stitched together. There's some variability. We talked about the benefits in the immune system of that. And anyway, you have to add a cap and a poly A tail for protecting the RNA from eroding. So those are things that are important from that processing. Hey, before I forget, you guys eat lunch and should leave here at 1:09. So if I get carried away, let me know. But today actually is not too bad. Excuse me. Let's see here. Number 3, Nathan.
The mRNA leaves nucleus and attaches to ribosome.
All right, remember, the ribosome has multiple components. It's made up of 2 subunits. Ribosomes are made up of 2 basic parts. What are they?
Protein and RNA.
Okay, proteins and rRNA. And there's 3 sites on the ribosome, the A, P, and E. The A site, what happens, Ashlyn? Yeah, so I'm going to make this go along with the letters. The A site, we'll call that the attachment site. That's where the tRNA attaches to the ribosome. Okay? The P site, Sarah, what does it do?
Attaches the protein.
Okay, so the protein there is made, the polypeptide is made, where amino acids are attached together and then are dropped off from the tRNA. And then the E site, Tay, what happens to it? E stands for, so the tRNA exits at that point. Okay, so attaches, proteins are made, exits. And so that's how proteins or amino acids are added to make that protein, okay, on the mRNA. You can have multiple ribosomes on one strand of mRNA. What's the benefit of that?
Makes multiple protein structures.
Yeah, you can make multiple proteins at the same time. If you look on page 353, you can have one strand of mRNA and you can be making multiple proteins at the same time. So it saves time and energy production of mRNA there. Number 4, Matthew.
Each amino acid attaches to its proper tRNA with the help of a specific enzyme and ATP.
Okay, so what type of bond is formed there when they attach?
Covalent bond.
Covalent, what type of covalent?
Peptide.
Peptide bond. Hence the name, growing polypeptide. Remember, that's the primary structure of proteins. Remember, structure equals function. And as we're going to see in the mutation sections today, if you do not have the proper primary sequence, then it's going to cause you some major issues in your life most of the time. So, yeah, I lost my train of thought. All right, sorry. On page 80 and 81 in your book, they go through the 4 different structures of proteins. So if you've forgotten those, you may want to go back and review. But primary structure is made by covalent peptide bonds where the amino acids are held together. The secondary structure, what happens?
They become alpha or beta.
Alpha helix or beta pleated sheets, which is held together by hydrogen bonds. The tertiary structure is folding. One of the main components there that helps them fold is disulfide bridges, which is a covalent bond that helps them fold back and forth. There are some hydrogen bonds and maybe even some ionic bonds in place there, but those disulfide bridges are probably the most important ones. And then in the quaternary structure, what happens?
Multiple tertiary protein structures.
Okay, so you have multiple proteins that attach together, like in the case of hemoglobin. We had the alpha and the beta hemoglobins, I think 4 of them to form that hemoglobin structure. We also talked about collagen. Lady, you probably hear about that. It's all the time in like lotions and whatnot, but you have multiple proteins that are woven together there that provide structure and stability to your beautiful face skin. Right, Ashlyn? Okay. And so that's the tertiary structure. So anyway, make sure that you're familiar with those. And number 5, Mariah.
A succession of tRNAs add their amino acids to the polypeptide chain as the mRNAs move through the ribosome, one codon at a time. When completed, the polypeptide is released from the ribosome.
What codes for the amino acid? Is the codon or anticodon? Codon. You always look at the mRNA codon when you're trying to code out whatever the amino acid sequence is on the amino acid chart, okay? Now, the tRNA has to what?
Transport.
It has to transport it. It also has to do what else? In order for it to drop it off. What else? It has to have what match up with the mRNA?
The anticodon.
So the anticodon has to match with the codon in order for it to drop off its amino acid, okay? So tRNA is important for that reason. It's got to match up. I want you to know how to complement. That's why I asked you to do that on the worksheet. But anytime you're looking at and you're coding actual proteins, which you may have to do on the test, then you always look at the codon on the mRNA, okay? All right, mistakes. Sometimes mistakes happen in DNA and protein production, and that causes problems. You can see here at the bottom of 355, we've got sickle cell. You guys may have heard of sickle cell disease or sickle cell anemia. Some people are just carriers of sickle cell. We'll talk more about that a little bit later on, and we get in genetics, and we spend quite a bit of time on this in the evolution unit. Keep in mind, evolution is going to be all about genetics and ecology mixed together, basically. How your genetics affects where you live and your survival. So anyway, Sickle cell. Well, let's talk in this context. Mutations, are they good or are they bad? Raise your hand if you think they're good. Raise your hand if you think they're bad. What do you think, Mariah?
I think they're bad mutations.
You think they're bad mutation? Miranda, what do you think?
I think it depends on what type of mutation it is.
They could be good, they could be bad, they could actually be both, and they could be neither. So, yes, it is very dependent on what is mutated. Okay? So, let's go through and look at the different types. I'm going to come back to this sickle cell chart here in a minute, but let's talk about what mutations are and look at the different types. Okay? So, on your notes, we got down to mutations, right? We talked about part 3. We went through genetic engineering, the reverse transcriptase, and all that. We discussed that yesterday.
We went through reverse transcriptase, and we got to like C.
Okay. Eukaryotes have introns. They have to be removed. Blah blah blah. That's all about the same. All right, mutations. A mutation is a change or mistake in the DNA sequence. Now, meaning it's not what it's supposed to be. When it's copied, there's some change. Or when something in the external environment causes it to change. So keep in mind, the structure of DNA is just made up by a bunch of different molecules. So you could have something outside influence that like affected it and caused it to break apart or some problem with that DNA. So that could cause the mutation. Part B, these are caused by mutagens or things that generate a mutation. Physical or chemical, or you could even say electromagnetic in nature, where you have like something around the electromagnetic spectrum like UV light from the sun. So anything that causes a change in that sequence. UV rays, what do they cause? Cancer. So UV rays, we can't see them. They're coming in. They're light waves. Sorry, from the sun, and so there's UVA, there's UVB, there's UVC. The ozone layer blocks out most of B and C, not most of A. A are more like causes your skin to be like wrinkly and old. Like you see those women at the beach and they've been laying out in the sun too much and their skin on their chest looks like a leather rawhide. You know what I'm talking about? Never mind. So, you'll know it when you see it. You'll be like, yes, you could make a leather belt out of that person's skin. Someone that's been in the sun way, way, way too much. Anyway, that ultraviolet radiation can damage the skin and cause cancer. There's 3 main types of skin cancer. I had a friend actually yesterday posted something that she had one on her face. She said she didn't even really know it was one or suspect it because it wasn't, I guess, significant in her mind. But she went and had it checked out, and sure enough, it was a kind of the least problematic cancer, basal cell carcinoma, or least problematic skin cancer anyway. So that can damage the DNA. We talked about in terms of cancer, there are 2 genes that can be modified and cause it. The 2 genes are what? You remember? Cause like 80% of all cancers. RAS and P53. Good. So RAS gene and the P53 gene cause 80% of cancers, and like I say, any of these could affect them and cause that. And I suspect, these are primarily the causes of most cancers. Cigarette smoke. They say cigarettes have about 43 different carcinogens in them, cancer-causing agents, things that could cause mutations in the DNA. And, a lot of people switch over to vaping. I'll be curious to see, we may not know for years, the effects of vaping and like how many cancer-causing agents are in there. Be interesting to see in the future as you guys get older. Y'all probably remember this conversation. It'd be like, read an article and it says 46 different carcinogens are found in e-cigarettes. I don't know. Maybe none. I doubt it though. C, alcohol in excess. I think that's like a way excess. Viruses. Viruses can cause DNA to change. We have a lot of viral DNA embedded in our DNA. Viruses embed their DNA in other organisms all the time. If you get a cold sore, it means you got some type of herpes virus on your lip, and maybe your lip was cracked. That virus got in the cell. The cell, it used reverse transcriptase to take its mRNA, turn it back into DNA, and embed it into your genome. And when it's cold outside and your lips get a little crack or it gets aggravated, then it triggers those cells to start replicating those RNA viruses and causes cold sores, and you have viruses that go everywhere. Don't put your mouth on the water fountain, Matthew. Don't do it. Okay. What else? Car exhaust. Do not breathe in the car exhaust. I think this is kind of a reference, to be honest with you, with just air pollution in general. For you people coming from apes, you three over there. We talked about the issues and different chemicals found in car exhaust, and so the more of that you have, the more likely you are to experience some issue. And then the last one, chemicals. That's kind of a broad range. Things like pesticides, poisons. These things can alter your DNA code. Really bad spider bites could alter your genetic code. Spider-Man, Walker, get with it. Okay. You people know what I'm talking about. All right, two major types of mutations. So, which one is worse, point or frame shift? Why? Okay, so it's modifying the whole sequence after where that mutation takes place. All right, so you might have stepped too late last night. Good. So point mutations. A point mutation is just a change in a single nucleotide, okay, which would affect a single codon. There are three different types of point mutations you need to be familiar with. A, Mason, go.
Silent point mutation. The mutation causes no change in the amino acid coded for. We would never know because it has no effect. This can happen because the codon coding is redundant. Remember?
Turn back to 339 real quick. Hold your place. So on 339, let's say that you are trying to code for serine, which is on that second column, SER. And let's say your genetic code, Walker, codes for UCU. But smoking too many cigarettes, those chemicals got to you, and you wound up having a mutation and change, and now it's CUA. What's going to happen? Mason? No. Sorry.
Yeah.
My bad. UCA.
It's like the lost country.
What's going to happen, Walker?
It's got to keep it as serine.
So, yeah, we talked about this the other day about the variability, because a lot of these have multiple codons for the same amino acid. And so if that's the case, you can have that mutation and it will not cause any problems. It's not going to show up. That's hence the silent mutation. Missense, Sarah?
The mutation changes the amino acid coded for. This is best seen in mutation that causes sickle cell.
Yeah, when you hear that term miss, like mistake, that means there's a problem. So this is one that's going to cause a change in the actual amino acid sequence. So on 355 in your book here, so sickle cell is resulted, it's the sixth amino acid. Not the sixth amino acid, the sixth, maybe the sixth amino acid. Yeah. The sixth amino acid, one of the nucleotides is changed from 5 to 3, from a T to an A, and which the mRNA code goes from GAG to GUG, And because of that, instead of coding for glucine, it codes for valine. Now, a hemoglobin protein is probably hundreds of amino acids long. If you have sickle cell disease, how many of your amino acids are changed out of those hundreds? 1. Why does only changing one amino acid cause such a significant shape change in the shape of blood cells? You've still got the same amount of amino acids.
Because changing the primary structure is going to affect.
Change the primary structure, which is going to cause
Changing the
secondary and tertiary structures. And so it's not going to fold properly. And you can see that like just changing one amino acid, one letter, one nucleotide, causing one amino acid to change is so significant. What are the problems associated with sickle cell? Tay? What? Blood clots. What else? Well, if your blood's clotting up, you're going to have less oxygen flow through, right? Pretend like this is a good example. Let's say you're on a lazy river. What are the shape of the floats that they put on the lazy river? Little circles that are concave, right? So basically they can squeeze and kind of bounce, and then they can keep moving through. Some capillaries are only really big enough for one blood cell to move through at a time. Now, these sickle cells aren't any bigger. They're just kind of a C shape. So why would that cause a problem? Well, they do hold less oxygen, yeah. And what else?
They can like get caught on something.
Yeah, they're basically like giant platelets. Or you can think of it like if you talk about a lazy river, you talk about a stream. You know, what happens when logs go down a stream or sticks? They get caught together and you have a log jam, and then it stops the flow. So that's basically what's happening with sickle cell there. Now, there is one main benefit to sickle cell, and does anybody know what it is?
Malaria.
Yes, so it infers some malaria resistance, not immunity, but resistance. But there are the drawbacks associated with blood flow. And it's pretty common. I want to say it's about 1 in 10 people of sub-Saharan African descent. So basically, like if you're black and your ancestors are from Africa, there's about a 1 in 10 chance of you being a carrier of sickle cell. I'm not sure. It's a recessive gene, so I don't know how often it shows up. Maybe it's like 1 in 100 or so to actually have sickle cell anemia. Yes?
I was gonna ask, like, what's the likelihood of you getting it as a Caucasian with no black ancestry?
It'd have to be probably some type of like spontaneous mutation. I don't know. Just circulating the population, it's probably very, very low. I know Hispanics, I want to say it's maybe like maybe I'm making this up, like 140 carriers. But you can be a carrier and just have some sickle cells, and you would call that having sickle cell, just being a sickle cell carrier. So you have some cells that are sickled and some most that are not. But sickle cell disease, like most all of your blood cells are going to be affected by that. So the same thing, like Hispanic people from India. There's some people that have it there, but it's much, much less than people of African descent. And I say sub-Saharan Africa because I would think the people who live in northern Africa of Arabic descent is probably a lot less too. Probably there, but it's much less. So even if it was 1 in 10 people, like if let's say you're black and like 1 in 10 people were carriers, you would have to have those two people have a kid, and then they would only have a 25%, a 25% chance of having a kid that had sickle cell disease. So that's why it's pretty rare to actually show up as the disease. Okay? We'll get more into that when we get to the Punnett square part though. All right, so I was just trying to show you how significant a change can happen just by changing one letter out of thousands probably. So, and then mutations. Some mutations can be good. Gosh, who was it? Somebody in first period was telling me about, I think it was Hunter. was telling me about something he had read and about those people in Nepal. There's like a certain type of soldier, and it's like Sherpas, but I think it was Gerpas or something. Maybe y'all know. I think this applies to probably Sherpas too. Y'all know what I'm talking about? The people that carry people up on the trips to Mount Everest. They have a certain gene that allows them higher oxygen intake and at higher altitudes. And so he was talking about the gene that was associated with that. Yeah?
I thought we talked about that last year in 855. I think that's a real thing.
Did we? No, I know it's a real thing. I just couldn't. He was actually, it was mentioned in the gene, and I can't remember what he said. But yes, so that would be a beneficial mutation. There are some mutations. What?
I read something about like a diving, like a it was a coastal village, I think in like India or something.
Oh yeah.
And they have like better eyesight underwater or something, like better vision for spearfishing.
Oh, I haven't seen that.
And they have they can also like hold their breath for like a lot longer.
Yeah, so imagine, I don't know if that's a mutation, well, the mutation or just natural selection. Like, let's say that you're better, like some people are naturally going to be able to hold their breath longer than others.
Right.
So, maybe the ones that don't hold their breath as well, maybe they die and they don't pass on their traits. So I wonder if that's just an evolutionary trait or if there's actually a specific gene helping them out. That's where we'll have to make the distinction. I know one time I used to show this video of these people from Italy. And, like if you think about going to the Olive Garden and eating lots of pasta and foods, Italian foods, I don't typically think of them as like being super healthy. And so, I don't know. Anyway, they found this, I'm not going to stereotype anybody, I'm on the internet. But they found these family in a village in Italy, and like they almost had like zero history of heart disease, heart attacks, or strokes.
Yes.
And they're, you know what I'm talking about? And they're like, that seems a little strange. And so they went in and investigated, and there was a gene that they had mutated that basically that gene created an enzyme that ate away most of the plaque buildup in their arteries and where it would normally cause a heart attack or stroke. And so even if they were eating the same normal meals as any other people in the area, but the heart attack and stroke was almost non-existent because of that. So I would say most mutations probably are bad because we typically have things in our body that are made certain ways to do certain things. And if you have a mutation, it's probably going to mess something up. But sometimes there are benefits. And like I said, the sickle cell one is beneficial and it can be harmful. Yeah.
Do you think like people that live closer to the equator or closer, to colder areas, do you think it's just evolution, or do you think it would eventually become like a mutation? Like them being adjusted to the cold or the heat?
I think that's just a function of evolution. So like typically if you think of Eskimos or what, I don't know if that's the right term nowadays. But typically, when I think of Eskimo, I think of someone who's a little bit shorter and has more like fat content, a little bit stockier. And I would think if you're closer to the poles, if your people evolved there a long time ago, that's more beneficial because more fat equals more warmth. But if you're leaner and skinnier, it's probably easier to move around and do stuff near the equator. I don't know. But some people naturally have genes where even if they live closer to the equator, they put on more weight when food's available. So like some people, like if they eat meals, they put on a lot of weight because their body's like, it's basically a gene that would say back in the day, like you don't have a lot of food very often. So when you do find food, we're going to hold on to it. But some people have very high metabolisms and they eat lots. And I guess that would be a function of their ancestors being around and having lots of food available all the time. We will get as in depth as I can on human evolution and why we're all a little bit different and how we're all very, very similar. We will get to that part, and I think it's all very interesting. And it reverts back to what you need to know about what you will need to know this from genetics. All right, so missense, there's a change. Nonsense. Who's the most nonsensical person in here? Go, Ashlyn.
an amino acid decoding for a stop codon, no protein will be made.
Ooh. So that means at some point during that sequence where it would normally code for an amino acid, it's a stop codon. So what's going to happen? It's going to stop. You're not going to make the right protein. You're going to stop short. And that's going to create some issues. I got 10 minutes left. So that's a big problem. That's probably the worst one out of the bunch there. All right, reading frame shift mutation where, now I'm going to make an analogy. Let's say you're taking a test, Mariah, and you're all wound up in the test and you look at a number and like the answer is D and you write it down, but you write it down in the wrong spot. And so you go back, you look at the next number and you get back on track and you just keep going. So the only thing that messes up is the one you wrote down wrong. Let's say you do something different though. Let's say you're on a test and you're just going one after another. You're not looking at the numbers and you skip number 11 and you go to number 12, but you write the answer for number 12 and number 11. And you do the same thing with 13, you write it number 12 and you keep going. What's going to happen at the end? Yeah, you're going to fail. So everyone after that point is going to be messed up. So that's how frame shift mutations work, okay? So it's going to mess up everything after that point. It's not where it's just messing up the one answer. It's messing up all the answers after that point. These mutations alter the codon sequence, the whole codon sequence after that point. Insertion, read that for us, Miranda.
Adding nucleotides to the sequence. For example, the big tan dog ran with inserted letter, the boy GTA, Indio, GRAN.
Yeah, so I like using the sentences here because I think it makes sense about how much sense it's not going to make to when you mess up this protein. So an insertion means a nucleotide is added in. So if your protein sequence was something like the big tan dog ran, okay, which makes sense, it's what it's supposed to be, but you added in an O, okay, you added in a nucleotide after that fourth one, between the fourth and fifth one, where it shouldn't be, it changes it so much. It goes from the big tan dog ran to the boy got a endo gran. So it's like it doesn't make any sense. It's not going to work. Everything changes. A deletion is basically the same thing, where you're taking out a nucleotide. So if we take out the I there, the big tan dog ran, you take out the I, the bigot and organ. See, that sounds a little different than the big tan dog ran. Okay? So those insertions or deletion causes major changes in the DNA sequence. Now, who does this affect and why? Part D, Mason.
Gametes and versus somatic.
All right, hold on a second. What are gametes?
Sex cells,
Sex cells. So sperm and egg, and somatic cells are
body cells.
body cells. Good. Go.
Gametes versus somatic. Who is affected? If a mutation occurs in somatic cells, the only one affected by the mutation is the person that the mutation occurred to. If the mutation occurs in gametes, the only one affected will be the organism created from that sex cell. This is how future generations may be affected, and this is a cause of evolution, changing DNA over time.
Okay. So this is a big deal. If your DNA in your sperm or eggs gets mutated, then it's going to have possibly affect your future generations, okay? Your offspring. Maybe something significant. If it affects one of your body cells, which is probably more likely because you got a lot more body cells than you do gametes, then it's going to affect you specifically, okay? So that's going to determine who it's going to affect and how it's going to be passed on. So anyway. 7 minutes here. All right, y'all got that math worksheet from yesterday. I think you people online have already done this. Tomorrow, we should be doing a lab in here, assuming that it comes in on time, hopefully. But just in case, if you guys will make sure you get the notes for the rest of the week are DNA control mechanisms, and biotech 1 and 2. I can assure you, the topics we cover are only going to get more complicated. So you need to make sure that you understand the concepts and things that we have discussed. And if you have trouble, you need to ask questions.